All right, so John Furber is going to go first. He's the CEO and founder of Legendary Pharmaceuticals. And actually, John, if you can go get a mic from Larry. He is a scientist and entrepreneur who's been studying the biology of aging, development, and regeneration for more than 25 years. He earned a Bachelor of Arts degree in physics and mathematics from the University of California in Santa Cruz in '75, and a Master of Science degree in biological sciences from the University of California in Irvine in 1990. Between degrees, he served the United States Congress as a technology policy analyst in the Congressional Office of Technology Assessment. He has a list of professional affiliations, which I won't go into detail on. He's a frequent contributor at meetings in the fields of aging, mitochondria, autophagy, and oxidative stress. These include Gordon Research conferences, Cold Spring Harbor conferences, et cetera, et cetera, et cetera. He has created several web pages that provide useful links for researchers and the lay public interested in aging, nutrition, bioinformatics, genomics, and molecular cell biology. For further details, he can give you his website once he gets out here. So let's welcome John Furber.
Okay, can you hear me? Not yet. Not yet. Well, it's a pleasure to be here. I first came to the Smart Life Forum in 2002, and now I'm living in Florida, so I don't get here very often. But it's good to be back. Now you can hear me. 1, 2, 3, 4.
You're fine.
Okay. So I've been to several aging research meetings in the last couple of months, and I thought I would just bring you up to date on a random hodgepodge of news. I think I'm ringing a little. Can you turn it down? Yeah. I thought I'd bring you up to date on a little bit of what's going on. Let me see if this thing advances. Yes. So I'm going to tell you some of the things that came up in the last couple of months at these three meetings. The Rejuvenation Biotechnology meeting was yesterday and the day before up at the Buck Institute in Marin County. The Harvard Medical School Paul Glenn Symposium on the Biology of Aging was in Boston the third week in June. And the American Aging Association had their annual meeting up in Seattle the first week in June. This was yesterday or the day before up at the Buck Institute, a beautiful location for a meeting. The meeting was organized by Aubrey de Grey and the SENS Foundation, and they held it up at the Buck Institute, and they're doing some collaborations up there. Quite a few people showed up. You can go to the SENS website, sens.org, and you can actually see some live video of the talks that were given at this meeting over the last couple of days. The keynote was by a man from University of Southern California, and his name is Pinchas Cohen, and he was talking about humanin, which is a peptide found in the mitochondria. And it protects from Alzheimer's disease-related neurotoxicity. It induces mitochondrial biogenesis, and the mitochondria, of course, help our cells to convert sugar and fat into ATP energy. And they did some measurements and found that children of centenarians have 3 times higher humanin levels in their cells than other people of the same age. And this is perhaps one reason why centenarians live so long. And it's possible that this can be used as a drug in the future. It's not currently anything other than an experimental material, although it's found naturally in our own bodies.
Sorry about that. I'm having every imaginable sound issue today. There you go. Try that.
Hello? Does it matter which way I turn my head?
Yeah, it does.
Left, right, left, right. Okay, so, oh, actually, I can look at my screen this way, and you can look over my shoulder. How's that? Okay. So in C. elegans worms, okay, now we're changing subjects because, like I say, I'm giving you some news flashes. Many of you know that a lot of researchers like to work with fruit flies, yeast, worms, and mice and draw conclusions about people from them. But they reported in this meeting at the Buck Institute that they are getting inconsistent results with their worms. They'll run the same experiment twice under exactly the same conditions, and in one experiment the worms will have a short lifespan, and in another experiment under exactly the same conditions the worms have a long lifespan. So they're having to go back and look at a lot of reports that have come out over the last 20 years and wonder whether they can really trust the results where they thought that some compound was good or bad for your lifespan. So some of the old worm research needs to be re-examined or maybe just chucked out altogether because of these inconsistent results. Also at the meeting was Gary Hudson, who started Elysium Biotechnology, which has a technology that kills senescent cells. Now, the older we get, some of the cells we have in our bodies stop dividing, and we say that they're senescent. Essentially, they're no longer dividing, and they're secreting bad growth factors. And it turns out from some experiments at the Mayo Clinic that in mice anyway, it's better off if they just kill those cells off. And then the mice actually live longer if they kill off their senescent non-dividing cells because they're no longer secreting these bad growth factors anymore. And this Elysium Biotechnology is a biotech startup here in Silicon Valley that's developing a novel therapeutic strategy for killing off these cells. And hopefully within the next couple of years we'll be able to kill off our own senescent cells. Meanwhile, you can take some readily available quercetin, and that kills some of the senescent cells, but not all of them. Then we had a talk by a philanthropist named Michael Grieve. He has a foundation called Forever Healthy, and that's the website, foreverhealthy.org. And he's one of the funders of Elysium Biotechnology, but he's also giving $1 million a year for 5 years to Aubrey de Grey's SENS Research Foundation. And he wants to help other entrepreneurs create startups to create therapies. So big hand for Michael Grieve. If he was here, I would clap my hands for him, but he's not here. Okay, let me see if I can advance. Yes. Solano Pharmaceuticals was started up by a woman researcher from Lawrence Berkeley Lab over in Berkeley, and she's been working for the last 5 years to develop a compound called XJB-5-13 that protects the mitochondria. She's done all of the mouse studies. She's ready to start clinical trials, but she needs to raise some money. So maybe she can go back to this guy, Michael Grieve, and raise some money from him to start clinical trials. It helps Huntington's disease mice, and she believes it will also help Alzheimer's disease, but that is yet to be 100% proven, except for the fact that it really helps the mitochondria, and if you help mitochondria, you're probably going to help all sorts of mental conditions. Some of you may get Life Extension magazine, and you may have heard of the Maharaj Clinic in Miami Beach in South Florida, and they may soon be offering young plasma infusions. And those of you that have been following aging research may have heard about some research that was done 10 years ago at Stanford where they took plasma from young mice and gave it to old mice, and the old mice got healthier. So the Maharaj Clinic in South Florida might soon be offering these infusions to people. There's also a clinic, and this wasn't announced at this meeting, but there's a clinic setting up in Monterey that's going to be doing the same thing. And there's also a clinical trial going on at Stanford with Alkahest Corporation. Alkahest is testing young plasma to see if it helps Alzheimer patients. But the clinic in Monterey, you just have to give them $8,000 and you don't have to have Alzheimer's to do it. They've just recently started operation down there in Monterey. Brian Kennedy is the president and CEO of the Buck Institute for Research on Aging, and he rattled off a list of things that look promising from their work on mice and smaller organisms. Rapamycin, for example, metformin, non-steroidal anti-inflammatories such as ibuprofen, something called acarbose, and then resveratrol, which is called a stack, which has to do with sirtuin activators. And almost everybody is now in agreement that one of the most important things you can do is exercise. And it's, if you're in really bad shape and all you can do is walk, well, you should walk. But if you can lift some weights, do some push-ups, some pull-ups, deep knee bends, the kind of resistance exercise does you more good than the aerobic exercise. But both kinds are good for you. Anything you can do that makes you breathe hard, sweat a little bit, you should be doing it. Everybody's in agreement now that exercise, don't overdo it at first. If you're out of shape, don't kill yourself, but just do a little bit and then do a little bit more gradually and work your way up. And also skipping at least one meal a day. It's called intermittent fasting. Seems to be very helpful for people as well as animals. Jean Loring, professor at the Scripps Research Institute, genomics-based quality control for stem cell therapy. She was looking at Parkinson's disease using human pluripotent stem cells that she makes from the person's skin using the Yamanaka protocol. And then they run them through their special quality control to make sure that the stem cells that they're making from the skin cells are not cancer. That's why quality control is important. So I thought her research is very important. We can't use it today, but they're starting to develop it down at Scripps. James Brown, not the singer, Vice President Corporate Development of Aldebaran, talked about a viral vector, and that means a piece of DNA that acts like a virus, but it can also carry other genes called plasmids into cells to treat cancer and blindness, which is pretty exciting. Once again, this is not yet available, but could be available soon. So the genes can code for messenger RNA that can be dosed in situations where just giving a protein therapy would not work. You give the mRNA and your own cells make the protein right where it's needed. He mentioned two companies that are getting started with this. One's called Moderna and the other's called Phase RX. Evan Snyder at the Center for Stem Cell and Regenerative Medicine core facility at Sanford Burnham Medical Discovery Institute in Southern California is taking umbilical cord cells and turning them into induced pluripotent stem cells. And they want to look at the gene expression of the induced pluripotent stem cells from centenarians and compare them with progeria patients and normal people and see if they can figure out what is it that centenarians have that normal people don't have and people with progeria maybe lack totally or vice versa. All right, what are the good genes and what are the bad genes? Now, last month at Harvard Medical School, there was the Harvard Paul Glenn Symposium on Aging. They have this once a year every June, and it's a one-day symposium. They don't charge any admission, and so it's really a nice philanthropy of the Paul Glenn Foundation to be funding a whole bunch of research. And there were some interesting developments announced there. George Church, and you can go onto this website, hms.harvard.edu/aging research, and you can see the program and some of the pictures and details from this meeting. George Church was a keynote speaker. He said, interestingly enough, age reversal is easier to test in clinical trials than life extension because you can do it faster. If you can take somebody and make them physiologically younger, you can see that right away. If you just hope that you're extending their life, it might take 20 or 30 years to get a result. So that's kind of an interesting flip on our thoughts, that maybe age reversal is easier than life extension in order to test. Also, age reversal can be tested in animals with new technologies. He also announced rapamycin, which was also mentioned by Brian Kennedy in the previous slide. Rapamycin extends animal lifespan and is now in human clinical trials. Jim Kirkland has been working with these senescent cells. I mentioned Osin Biotechnology, which is working to kill senescent cells. But before there was Osin, the Mayo Clinic was killing senescent cells with a cancer drug and with the quercetin nutritional supplement. And he said in some ways, senescent cells are like early stage cancer cells that don't divide. In other words, they secrete these bad things, and that's why a lot of times you'll see cancer patients, they have cachexia. Their muscles are dissolving. And so it's not just the rapid division of cancer cells. Cancer cells are actually toxic to the body. And they fail to turn over their misfolded proteins in autophagy. And they have a Warburg shift similar to cancer cells. So once again, he, like all the other scientists, say exercise and partial day fasting are good for you. Rafa Di Cabo has been a longtime researcher at the National Institute on Aging. He pointed out that in contrast to what a lot of newspaper reporters will say, caloric restriction doesn't work in all strains of mice. You probably have read all of these headlines. Caloric restriction works in every animal that's ever been tested. That's not true. They took 50 different strains of mice. They put them into control groups and caloric restriction groups. And about a third of the strains of mice lived longer. About a third of the strains of mice lived shorter. About a third of the strains had no effect at all. So you can't always believe the newspaper headlines. These are just aging cell and experimental gerontology have scholarly refereed papers showing that. But he also agrees partial day fasting is beneficial. And the third meeting I'm going to tell you about is the American Aging Association, which has an annual meeting in a different city every year. And this past June, they had their annual meeting in Seattle, Washington, right on the waterfront. Oh, that's the wrong website. Anyway, so ignore that website. That's from the Harvard meeting that I just told you about. But Bess Frost from the Barshop Institute, identification of Alzheimer's disease as a neurodegenerative laminopathy. Now, the nuclear lamina is the envelope that surrounds the nucleus of the cell. And apparently in Alzheimer's disease, she and her team have recently discovered that there are little holes in the nuclear lamina that shouldn't be there. And it's the lamin B protein. And these pores allow cytoplasmic factors to go into the nucleus and nuclear factors to come out into the cytoplasm. And that this is one of the pathologies of Alzheimer's disease that had not previously been reported. So that's kind of giving us some ideas of some new research that might result in new therapies a few years from now. At any rate, it results in a larger amount of messenger RNA being exported from the nucleus, and then this phosphorylated tau tangles gets into the nucleus and messes up the nuclear function. I mentioned the Maharaj Clinic in South Florida. Life Extension Foundation, in one of their magazines a couple of months ago, mentioned that they are raising money for a study of young plasma transfusions at the Maharaj Clinic, which you can read about on the lifeextension.org website. But as I say, you can just drive down to Monterey and do it. You don't have to go to Florida. And then infusions of human, so both the Maharaj Clinic and the Monterey Clinic and the Stanford clinical trial have been working with blood plasma from 20-year-olds, 20 to 29-year-olds, who have the young factors that us 60-year-olds don't have. But some of the unpublished research is looking at umbilical cord plasma, which seems like it might have even better effects. But this is not yet published, and it may or may not prove to be any better than 20-year-old plasma. But it's just something I thought I might mention to you to keep an eye on. Next, the Salk Institute in Southern California has been working for 5 years with a compound called J147 that improves brain function. And recently they modified it slightly, and they call the modified version CAD-031. And this looks like it's going to be a real winner. I'm very excited that I'm going to find a way to get my hands on some of it and do some experiments with it. And finally, there's another compound called SS-31 from Stealth Pharmaceuticals that improves mitochondrial function. And if you improve mitochondrial function, you should be able to help the heart, the muscles, and the brain cells. So that's one that's also worth keeping an eye on. And it's also something that I'm hoping to get my hands on and do some personal experimentation so I can report back to you guys. Dudley Lamming at the University of Wisconsin has been looking at maybe a high-protein diet not only isn't helpful, but might be actually harmful compared with a low-protein diet. And this sort of turns a lot of old research on its head, because the old bodybuilder mentality from 20 years ago was eat lots of protein, don't eat any starch, don't eat any carbohydrates. But he's finding that if you limit your protein, it helps you to limit the amino acid methionine, and apparently a low methionine diet is healthy, and if you just cut down your protein, that's the easiest way to limit your methionine. So what should we eat? He's another proponent of partial day fasting and caloric restriction, but he says that's hard for people to do. So he's very interested in finding small molecule drugs that might provide some of the benefits of fasting without the willpower. Myself, I think I'd just save the money and skip breakfast. So what is altered in a caloric restricted diet? There are 3 macronutrients: proteins, fats, and carbohydrates. There are good fats and bad fats. The omega-3 fatty acids are generally good. Bernt's going to disagree with me when I say saturated fats are bad, but I'll let him defend himself. Many of the past recommendations were we need to eat more proteins. But Cell Metabolism 2014, Solon Biet is the author. The low-protein diet extends lifespan in mice compared with higher protein but the same total calories. That's what isocaloric means. And the human diabetes risk is cut in half with low-protein diet. And I was just totally astonished by that. Like, we always thought, it's the sugar that causes type 2 diabetes, but apparently high protein can be bad for you. He collaborated with Luigi Fontana at Washington University in St. Louis. They looked at protein restriction in people for several weeks. These people on the... Is that me?
Yeah, that's okay. You're not doing it.
Okay. They looked at protein restriction in people for several weeks, took their blood, measured things, and they found that these people lost significant fat weight, adipose weight, by cutting their protein intake. And they also had a decline in fasting blood glucose levels from cutting their protein intake. Protein promotes satiety, so the low-protein group actually ate 10% more calories and lost weight and lost fat. And this is just astonishingly new news to me. I guess that's the end of my slide. I guess that was my last slide. Okay. If you have questions, I may or may not be able to answer them, but I'll try. You had your hand up first.
Protein is acid. There's another people looking at alkaline versus acid. Is that just a simple way of saying have more alkaline diet and less acid. And that also then, we don't have to talk about the protein, but just have alkaline food.
No, it doesn't seem to be the case. It seems to be actually the protein, even though protein is made of amino acids, because they looked at all sorts of different combinations, and they found lower protein, particularly methionine-containing proteins, would seem to be the culprit, and it wasn't just acid versus alkaline.
When you say low protein, what are you talking about?
You'd have to read the papers that I referenced, because I'm not an expert in these things. I just attend the meetings and take some scribbled notes.
And when you say low protein, do you replace it with something else, or is it just lower calories?
No, in this experiment, because the people on the low protein diet were hungry, they actually ate 10% more calories than the control group.
I've been looking at the protein, and I spoke to the gentleman who did the study, and one of the things I introduced to him was when I was working with Roy Walford on the caloric restriction diet. The reason caloric restriction diet works is because you're reducing mTOR pathways. That's what rapamycin does.
Yep.
Spoke with John Miller about that, who is one of the key persons behind rapamycin, you know, who's done all the studies. And they agreed that it's the type of protein that you put in your body that has more of a problem than what you eat. So by reducing methionine, number one, tryptophan, number two, and cysteine, you reduce mTOR pathways and leucine. So that's why I got into collagen. Collagen is the only protein on the market that is deficient on all levels of methionine, tryptophan, cysteine, and leucine. So as you get into 65 and older, this studies just came out of New Zealand that people at that level have to be on a higher protein diet because of the factors of muscle loss and everything like that. So a big factor of rapamycin is really reducing mTOR. So the big factor that all the scientific papers and all the universities are doing now, they're looking at mTOR pathways, reducing that, and IGF-1. Those are the key two molecules that they're looking at for aging mechanism, restoring longevity, and mitochondria, right?
Well, Bernd, you certainly look healthy, and you're older than me, so.
The other thing about, I've been on the phone a lot with Bruce German out of UC Davis and Robert Ward out of Utah. They're doing, and there's a guy named Robert Veach. Look him up. He's a world-renowned ketogenic expert. He's probably the best in the field. And they're all now shown that their science has shown that certain types of fats that we need to consume, like saturated fats from butter, cream, MCT oil, coconut oil, and I think Steve will agree with me because he's done a lot of work in that area. There's now a study just came out of UCSF, showed that low cholesterol diets leads to cataracts, and I told you that over the phone. And there's a lot of papers on that. And in Denmark, Sweden just recently published that low-fat diets are shown to have higher LDLs, lower HDL levels, and also are shown to have more obesity problems now and depression and suicidal tendency. This is a published paper by Denmark and Sweden.
Yeah, so that sort of goes along with these people in the last paragraph. They were eating less protein, but they were actually eating more fats and carbohydrates.
Yeah.
And they lost fat, and they lost weight, and they were very healthy.
Did they make any differentiation between animal protein and plant protein?
They did not.
No.
Any news on Tree Halos for autophagy?
Nobody talked about it.
What's the latest?
I don't know. Tree Halos.
Oh, Tree Halos. That's a mushroom derivative. I take it every day. It's supposed to induce autophagy in some ways in certain amounts, and it's found in mushrooms.
How much do you take?
I don't measure anything I do. I just take it.
Like a cup?
No. Maybe a teaspoon to not 1 or 2 teaspoons. But it's an
do you eat the
interesting
whole
molecule.
mushroom, or do you eat an extract?
No, I get it from a major pharmaceutical company that sells Oni Tree Halos.
Okay.
Yeah, I read a lot about it, and it's very interesting. And I've even seen where they actually put it in the eyes, and it's restored some of the function of the eyes through the lens and all that.
And was there a question back here?
What was that called?
TREHALOSE, right?
My question just relates more to this intermediate fasting, the frequency of something like that. Is that something that's I wouldn't imagine to be an everyday thing. It's probably based on someone's BMI and such like that. But
Just skip breakfast and lunch a couple of days a week, and you'll notice some benefits. Because
and
if you go for more than 12 hours without, so if you have coffee or tea in the morning, have it without sugar and cream. So you're not getting any calories for at least 12 to 14 to 15 hours. And that turns on your autophagy, which helps your cells to tear up and digest the misfolded proteins. If you constantly supply your cells with food, they never recycle the damaged stuff. But if they're a little bit hungry a couple times a week or every day, they will recycle the damaged stuff and your cells will be healthier.
And at what point in that cycle does the body start to consume fat versus muscle? Like I don't even, it's probably not a moot point, right? I mean, it's probably not going to start taking muscle, depending on someone's composition.
Well, see, I don't count calories. So I skip breakfast and lunch and then I eat like a pig at dinner. So I'm not starving.
I just want to add a comment or two here. It may be a little out of line, but could I ask how many here in the audience have heard of Joel Wallach, the nutritionist, the scientist, Nobel Prize nominee? Well, anyway, he says lowering the amount of cholesterol that you take in, which all the physicians all advise, is the primary reason for Parkinson's, mental decline, Alzheimer's, and it's a physician-caused disease. He says to eat 6 eggs, as many as you want, have all the cholesterol, you'll be on the right track for longevity. He also says the reason why we don't live our full potential is because of nutritional deficiencies, the lack of the minerals in the soil, which have been uptaken and is not there anymore. So he suggests what he calls 90 essential nutrients, essential fatty acids, vitamins, minerals, and the other bunch that he promotes. And there are many people that have taken to his program, and that's my comments.
Thank you.
Any other questions? What's the
Microphone.
latest? Hold on.
Wait for the mic so that they can get it on the internet.
Hi. Did anyone mention the product from Elysium, the basis, the NAD?
Nobody mentioned it. I've heard that name.
Yeah.
Maybe you can tell us.
I just saw it recently, and I don't know much about it. I read a couple studies that were in Science.
And did it look good or bad?
It looked good. Yeah, it was mouse studies, I think. But it improves mitochondrial function. So
is that the one with the blueberry extract, the pterostilbene? Yeah. No, it's not. Well, it's resveratrol plus pterostilbene.
Guarante is using right
Lenny
now.
Guarante, yeah.
Yeah, he's using stilbene from the blueberry extract, and he's also using resveratrol form.
Yeah.
And NR, which is from chromadex, which is nicotinic D-ribose, a molecule found in milk.
So I think it's probably good stuff. He's a good scientist. He's got a good scientific team with him, but there might be cheaper ways to get it because that stuff's kind of pricey.
Yes.
There are some scientists who believe that simple nicotinic acid, which is a form of niacin, might be as effective as this expensive NR. And you can get pterostilbene maybe from some other sources that are a little cheaper. So you're not gonna go wrong if you buy the high price stuff. But if you're on a budget, you might want to look around at some alternatives.
Well,
Would you
there's
agree,
still-
Bernd?
Yeah, I agree with you 100%. Low dose of niacin, 100 milligrams several times a day, or niacinamide. I take 1,000 milligrams in the morning, and Ray Pete is very strongly behind niacinamide as just an NAD, increasing NAD levels, you know.
Well,
And-
apparently the nicotinic acid form of niacin and the niacinamide forms are metabolized slightly differently, so
Yeah.
you might want to take a little of each.
Yeah, but make sure you don't take a large dose of niacin, because then you get the nitric oxide flush, which is something you don't want. So this cardiologist who I've been talking to a lot, he recommends no higher than 100 milligrams. And I think you agree with that too, right, Stephen? A low dose niacin with, so you don't get the flush, and it works and converts to, you know, NAD much more efficiently.
Now, I've actually been taking the niacin every morning, 500 milligrams, and I love the flush. I feel like it's irrigating my skin and giving it extra blood, and so I get it out of the way in the morning, and then it's like I've just run a mile or something. My face is all flushed, so I kind of like the flush.
And if you take it multiple times during the day, each subsequent dose produces a lower flushing effect. And I'm not convinced that a lot of things that, if done continuously, that can be adverse influences are necessarily that way if you do them more judiciously. So a short-term spike in nitric oxide doesn't bother me as much as having chronically elevated nitric oxide. So
I don't do the time release.
There's a lot of stuff like that as well in terms of like exercise. You know, if you over-exercise, it's quite deleterious.
Right.
And so, a lot of things in moderation, and likewise fasting in moderation as well.
Mm-hmm.
And so I suggest that people not only do the daytime type of fasting of skipping breakfast and then skipping breakfast and lunch occasionally, but periodically to do a 24-hour fast as well, maybe even a little bit longer, but not to do it 3 days or longer.
Yeah, I would agree with all of that.
Can one of the speakers, Steve or our guest speaker, clarify the differences between niacin, nicotinic acid, NR.
Niacinamide.
Niacinamide.
And nicotinamide riboside.
Yes, and NADH.
Well, I'm not going to be able to tell you verbally, but it's something you can look up. They're just slightly different molecules.
Yeah, they're slightly different molecules that undergo different synthetic pathways on their way to make complex 1, or otherwise known as NAD or NADH.
Yeah.
And the difference between NAD and NADH is merely that one is oxidized, the other is reduced, and they're constantly converting back and forth between each other. So companies, companies that really hype that issue, you have to take it as NADH, or NAD is better, are probably hyping their product.
They're trying to create a marketing distinction.
The problem with NAD is that it's difficult for the body to make it. Okay, you can make it from niacin or niacinamide, and it's an incredibly inefficient process. Or you can make it from tryptophan, which I think you get about 2% yield for every gram of tryptophan. So that's also a very, very awkward pathway to make it. So the idea that this might be beneficial to supplement it, I think, is very useful to explore. And I suggest that this is a good self-care experiment to try on yourself, to do a period of supplementing NADH, because that's probably the most complete supplement that you can get, where the entire synthetic pathway is pre-provided for you. And just see, does it help your sleep? Does it help your strength? Does it help your stamina? Does it help your heart, your mental performance? And that maybe you're going to spend $40 in doing that experiment, but I think that it's nice to identify yourself if you are an NADH responder.
And it might be interesting in a couple of months if some of you try various experiments to report back to us.
One of the things that I noted when I was publishing the SERI newsletter was that most people are non-responders. They cannot tell the difference when they're taking NADH. And maybe 1 out of 20 will, in the general population, will respond positively to it. But among Parkinson patients, it's more like 40%. So it could be that people who have some kind of an energy lesion in their metabolism, it's because of a problem with NADH or NAD.
Okay, let's wrap up here. Let's give
Okay.
John a round of applause. Thanks, John.
It's been a pleasure. Thank you.